Hi, so we'll start the class now. So today is the 11th problem solving session for the course overview and integration of cellular metabolism. I hope my voice is audible.
Yes, ma'am. It's clear, ma'am.
Yeah. So, as you know, the course instructors are Professor Aritri Beer and Professor Arindam Ghosh from IIT Kharagpur, and I'm Shrija Ghosh, a PMRF from IIT Kanpur. I'll be helping you with the assignment questions related to the 11th week's topic. So today we'll be discussing questions related to purine metabolism disorders, pyrimidine metabolism and its related disorders, and then uh, some other inborn metabolic disorders, as well as we'll now see whatever metabolic pathways that we have studied till now. We'll see the overall integration, what the course title actually is: overview and integration of cellular metabolism, right? So till now we have read each and every metabolic pathway individually. We read the metabolism of carbohydrates, then metabolism of proteins, then fats, and then metabolism of nucleotides, which you are going to study now. So, after studying all the pathways individually, now we have to see how these pathways are integrated and occurring simultaneously in our body. Like, it's not like in one time, like in at a moment only carbohydrate is getting metabolized and nothing else is happening, or only fats are getting metabolized. Everything is happening together inside our body, inside our system, right? So how everything is connected to each other, and how all the metabolic pathways are integrated uh, with one another, we'll see that. So what happens when we are uh, eating, and what happens if we are not eating, if we are under condition of starvation. So in those cases, what is happening? So all those conditions. So this will basically be a kind of a revision also. So because we'll be revisiting all the metabolic pathways that we have studied till now, and you will have a better understanding of all these pathways, and how they are actually have occurring in our, like how they are actually functioning in our inside our body or inside our system. Okay, so I'll start with the questions. Feel free to ask any doubt you have. Just unmute and ask, or write it in the chat box, whichever way you are comfortable. So I'll start with the questions. You can write the answers in the chat box, and we'll just have the discussion on that. Okay. Okay. So starting with the first question, which molecule serves as a common precursor for both purine and pyrimidine nucleotide biosynthesis? Which molecule serves as a common precursor for both purine and pyrimidine nucleotide biosynthesis? Option A, AMP. Option B, PRPP. Option C, uric acid. And option D, hypoxanthine. Which molecule serves as a common precursor for both purine and pyrimidine nucleotide biosynthesis? Option A, AMP. Option B, PRPP. Option C, uric acid. And option D, hypoxanthine. You are saying option B, that is PRPP. That is the correct answer. So we have discussed this in the previous session also, when I have discussed purine biosynthesis. So PRPP is a common precursor for both purine and pyrimidine biosynthesis pathways. Basically, PRPP is phosphoribosyl phosphoribosyl pyrophosphate. So this PRPP actually donates the ribose group in the uh, purine or pyrimidine nucleotide. Okay. So a nucleotide contains a base and a ribose sugar and a phosphate group, right? So that makes it a complete nucleotide, right? Here you can see it's AMP. So adenosine, ribose, phosphate. So one phosphate is there, that's why monophosphate. ATP has three phosphates, okay? And the ribose is coming from ribose 5-phosphate. So from ribose 5-phosphate and 1 ATP, with the help of the enzyme ribose phosphate pyrophosphokinase, that is PRPP synthetase. It is also known as PRPP synthetase. I have mentioned in the previous sessions also that when the name synthetase is there, ATP hydrolysis is going to happen. And in this case, 1 ATP is hydrolyzed into AMP. That is two phosphate groups are removed from the ATP. That means the energy required to break two phosphate bonds or a pyrophosphate bond is required. So it's a high energy demanding reaction, and the resultant product that we get is phosphoribosal pyrophosphate, which is the precursor for donating the ribose sugar in both purines and pyrimidines. And that's why it's numbered as step 0, because this is not the first. So in case of purine synthesis, you will see that this is the first step, but then since this is a common step for both purine and pyrimidine synthesis, we don't label it as the step 1, but we label it as step 0. And then from here, the next step would, for purine synthesis, would be the step 1. And for pyrimidine synthesis, we'll have a different sequence of steps. We'll discuss it later. So this is a preparatory step or step 0, where PRPP is synthesized by the help of the enzyme PRPP synthetase or ribose phosphate phosphokinase. Okay. So ribose 5-phosphate with the help of ATP forms phosphoribosal pyrophosphate, where it takes up two phosphate groups from the ATP and ATP gets converted to AMP, that is adenosine monophosphate. Okay.
Okay, ma'am.
Moving to the next question. Deficiency of HGPRT leads to which clinical syndrome? Option A, gout. Option B, Lesch-Nyhan syndrome. Option C, ornithine transcarbamylase deficiency. Option D, maple syrup urine disease. Deficiency of HGPRT leads to which clinical syndrome? Option A, gout. Option B, Lesch-Nyhan syndrome. Option C, ornithine transcarbamylase deficiency. And option D, maple syrup urine disease. You've answered option B, that is Lesch-Nyhan syndrome. That is the correct answer. So Lesch-Nyhan syndrome, it is a metabolic disorder caused by the deficiency of an enzyme HGPRT or HPRT. So this is the enzyme that is sorry. This enzyme HGPRT, which is also called hypoxanthine guanosine phosphoribosal transferase. So this enzyme basically is the key enzyme in the purine metabolism, and it converts the hypoxanthine and guanine into the corresponding monophosphate. So basically, it converts the hypoxanthine into IMP and uh, guanine into GMP. Okay. And this enzyme is involved in the salvage pathway of the purine synthesis. So all this has been discussed in the previous session, so I'm not going through it once again. So Lesch-Nyhan syndrome, it is a metabolic disorder caused by the deficiency of this enzyme HGPRT produced by mutation in the gene located on the X chromosome. The disease is marked by the buildup of uric acid in all the body fluids that results in condition known as hyperuricemia or hyperuricosuria. Uh, why this is happening now? I've told you that HGPRT is responsible for the uh, I've told you HGPRT is responsible for the conversion of hypoxanthine and guanine into its corresponding monophosphates, right? Now, if that enzyme is deficient, that means there's going to be accumulation of hypoxanthine and guanine, and all these intermediates of nucleotides biosynthetic pathway, they, if they are accumulated, they enter the uric acid uh, cycle or urea cycle. So in that case, there is a high buildup of uric acid in the body fluid. So uric acid in blood will lead to hyperuricemia. Uric acid in urine will lead to hyperuricosuria. Okay. So buildup of uric acid occurs because all the intermediates of the uh, nucleotide biosynthetic pathway, they enter the uric acid cycle. Okay, and the symptoms, because of the uric acid buildup, the symptoms include severe gout. Gout is basically the deposition of uric acid at different joints. So gout formation occurs. So that's a major symptom in this syndrome. Then impaired muscular control, moderate mental retardation, and kidney problems. So, all these are symptoms. These complications frequently emerge in the first year of the life. Neurological symptoms can include facial grimacing, involuntary writhing, aggressive behavior, self mutilation, and repetitive movement. So, patients are many times patients are observed to self mutilate themselves, so they are observed to bite their fingers off or scratch their skin. So those kind. So all these are neurological symptoms. The since there are symptoms related to mental retardation, and then that basically our central nervous system is affected due to the uh, deficiency of this enzyme. Okay. Think of it in this way, that if a GPRT is not there, the uh, nucleotide biosynthetic pathway will be hampered, the intermediates will be accumulated, and there obviously nucleotide is the basic uh, requirement of life, right? So it is the basic building blocks for the DNA, and DNA are the basic building blocks of any organism. So if you think of it in that way, any defect in the nucleotide biosynthetic pathway is going to have an effect on your central nervous system. Okay. So the mental deficits in behavior do not react well to therapy. There is no known cure, but many patients can survive till adulthood. So there's no cure for the syndrome as well. Okay. On to the next question. That's this is clear, I hope.
Yes, ma'am.
Okay. Third question. What is the final product of purine degradation in mammals? Option A, hypoxanthine. Option B, xanthine. Option C, uric acid. And option D, allantoin. What is the final product of purine degradation in mammals? Hypoxanthine, xanthine, uric acid, and allantoin. What is the final product of purine degradation in mammals? Hypoxanthine, option B xanthine, option C uric acid, option D allantoin. You are saying option D allantoin. The final product for purine degradation, I just discussed, like not exactly this, but I've told you the where the intermediate of purine biosynthetic pathway goes, right? So yeah, option B is also not the correct answer. Actually, the correct answer is option C, uric acid. So intermediates of purine biosynthetic pathway like hypoxanthine and xanthine, they all enter the uric acid cycle. So similarly, when purines are degraded, they they are also degraded into hypoxanthine, xanthine, which are its uh, starting point, right? Degradation will happen in the opposite direction of the biosynthetic pathway. So hypoxanthine, xanthine, and all these intermediates, they can enter the uric acid cycle. Okay, they can form uric acid. So the biosynthetic pathway goes in the other direction where hypoxanthine and xanthine are converted into IMP, XMP, GMP, which are the nucleotides, purine nucleotides, right? Now, if we are looking at the catabolism of purines, that is the degradation of purines, it is going to be in the opposite direction. So AMP, IMP, XMP, GMP, all these are purine nucleotides. So all these will be degraded by nucleotidase first to remove the ribose and the phosphate. So you get the purines. So adenosine, inosine, xanthosine, and guanosine. So these are the purines, just the purine ring without the ribose and the phosphate. Now these bases are then degraded into inosine. So adenosine is converted into inosine by the enzyme adenosine deaminase, and inosine is converted into hypoxanthine by the enzyme purine nucleoside phosphorylase, where inosine is sorry inosine is uh, trans converted into hypoxanthine. Then xanthosine is converted into xanthine by the same enzyme purine nucleoside phosphorylase, and guanosine is converted into guanine. Hypoxanthine and guanine then get converted into xanthine. So the whole pathway converges to xanthine, and xanthine is converted into uric acid by the enzyme xanthine oxidase. So xanthine oxidase can convert xanthine into uric acid. It can also convert hypoxanthine into xanthine. Okay, so xanthine oxidase can bind to hypoxanthine or xanthine to form uric acid. Okay. And xanthine oxidase contains FAD, molybdenum, and iron. So it's a interesting enzyme having the FAD as the cofactor, as the electron carrier, molybdenum, and iron. Two metal ion centers are there. Reaction, and this reaction produces H2O2. So it produces reactive oxygen species. So xanthine oxidase reaction, you can see in both of these steps are producing H2O2. Okay. So here you can see one H2O2, here you can see the second H2O2. Okay. So basically this is the degradation of purines, and the final product of degradation of purines is uric acid. Okay. Is this clear to you?
Yes, ma'am. It's clear now, ma'am.
Okay, then moving to the next question. Question 4. Which enzyme catalyzes the committed steps of de novo purine synthesis? Which enzyme catalyzes the committed step of de novo purine synthesis? Option A, glutamine PRPP amidotransferase. Option B, xanthine oxidase. Option C, adenylosuccinate synthetase. Option D, HGPRT. Which enzyme catalyzes the committed step of de novo purine synthesis? Option A, glutamine PRPP amidotransferase. Option B, xanthine oxidase. Option C, adenyl succinate synthetase. Option D, HGPRT. Which enzyme catalyzes the committed step of de novo purine synthesis? Glutamine PRPP amidotransferase, xanthine oxidase, adenylosuccinate synthetase, and HGPRT. What would be the correct answer? You are saying option A, that is glutamine PRPP amidotransferase. That is the correct answer. So this is the step 1 and the committed step of purine biosynthesis. So I told you the step 0 was the PRPP synthesis. So from PRPP, uh, we get 5-phosphoribosyl 1-amine, that is PRA, and this is the committed step. Why it is called the committed step? Because this is the step which is regulated by feedback inhibition, and this is a critical. Basically, this enzyme is uh, regulated tightly. This enzyme is tightly regulated in the whole purine biosynthesis pathway. So glutamine phosphoribosyl pyrophosphate, that is PRPP, is converted into glutamic acid plus pyrophosphate plus 5-phosphoribosyl 1-amine, that is PRA. So you can see here the structure. PRPP is there where pyrophosphate group is there, and then we have ribose 5-phosphate. So 5-phosphoribosyl part is there, a part remains, pyrophosphate is released, glutamine gets converted to glutamate, and the amine group is transferred at this position where the pyrophosphate is there. So we get 5-phosphoribosyl 1-amine. So amine group is transferred here. Glutamine gets converted into glutamate, and the pyrophosphate is released from the first carbon. Okay. So this is the purine this is the start of the purine biosynthesis, and the purine ring is assembled on the ribosyl 5-phosphate. So ribosyl 5-phosphate is already there, and the purine ring is assembled here. Okay, on this amino group, the rest of the synthesis will start. Okay, so this is the first step and the committed step of purine biosynthesis. This is the de novo purine biosynthesis pathway. Okay, I hope that's clear. I'll move on to the next question. Question 5. Allopurinol inhibits which enzyme? Option A, glutamine PRPP amidotransferase. Option B, xanthine oxidase. Option C, aspartate transcarbamylase. And option D, thymidylate synthase. Allopurinol inhibits which enzyme? Glutamine PRPP amidotransferase, xanthine oxidase, aspartate transcarbamylase, or thymidylate synthase. We are saying option B, that is xanthine oxidase. That is the correct answer. So allopurinol is basically analog of hypoxanthine. So you saw in the catabolism of purine, where hypoxanthine is converted into xanthine and then uric acid by the enzyme xanthine oxidase. So since these two are analogs of each other, you can see the structure of hypoxanthine and allopurinol. They are very similar. The only difference is of this nitrogen uh, and position of this nitrogen. And since the structure is very similar, allopurinol can actually inhibit the xanthine oxidase enzyme competitively. So it can fit in the active site where hypoxanthine actually binds, but binding of hypoxanthine will result in the production of xanthine, whereas binding of allopurinol will inhibit the enzyme. And these two, so this is the substrate, hypoxanthine, and allopurinol is the inhibitor, and this is a competitive inhibitor. It can compete with the substrate for the same active site. Okay. And this allopurinol is used as a common treatment for gout because gout is basically the accumulation of uric acid, right? So if hypoxanthine is higher in if hypoxanthine is higher and xanthine oxidase is present, then xanthine oxidase will keep converting hypoxanthine into uric acid, and then it can lead to gout. So to treat gouts, uh, xanthine oxidase can be inhibited so that hypoxanthine and xanthine will be there in the circulation, and hypoxanthine and xanthine, since they're soluble, can be excreted in the urine. So hypoxanthine and xanthine, they can be excreted in the urine if not converted to uric acid by xanthine oxidase. So in case of gout, xanthine oxidase can be inhibited by allopurinol, which is an analog of hypoxanthine, and allopurinol can actually be used as a treatment strategy. Okay. I hope that's clear. Let me know if you have any doubt. Moving to the next question, question number 6. In pyrimidine synthesis, what is formed first? Ribose phosphate group, UMP, pyrimidine ring, or PRPP? In pyrimidine synthesis, what is formed first? Option A, ribose phosphate group. Option B, UMP. Option D, pyrimidine ring. Or option D, PRPP. In pyrimidine synthesis, what is formed first? Option A, ribose phosphate group. Option B, UMP. Option C, pyrimidine ring. Option D, PRPP. You are saying option B, that is UMP. That's not the correct answer. So the correct answer actually is option C, that is pyrimidine ring. So if you recall the purine biosynthetic pathway, there the PRPP was the first thing that was forming, and from PRPP, the purine ring synthesis was going to occur on the ribose sugar, right? But in case of pyrimidine synthesis, first the pyrimidine ring forms and then the ribose sugar is attached from the PRPP. Okay, so this is the whole pathway of pyrimidine biosynthesis, where it starts with glutamine. So raw materials for the pyrimidine synthesis is glutamine, CO2, and aspartic acid. So we have glutamine, which is converted into carbamoyl phosphate. Then from carbamoyl phosphate, carbamoyl aspartate forms. So you can see it's still a linear structure, and then cyclization occurs once carbamoyl aspartate forms. It becomes dihydroorotate and then orotate. So orotate has the pyrimidine ring structure. Sorry. So orotate is the pyrimidine ring structure. And then the ribose gets attached to this pyrimidine ring structure to form the OMP and then later UMP. So UMP is the final pyrimidine, uridine 5 monophosphate. It is one of the pyrimidine. And from UMP, the other pyrimidines can also start forming. Okay. So the point is, in pyrimidine biosynthesis, first the pyrimidine ring forms, unlike in case of purine where the ribose, where it was starting on the ribose sugar. So the ribose PRPP was there, and on the PRPP, the purine ring was forming, right? If you have any doubt, you can go back to the previous session slides and see how purine synthesis takes place. So purine synthesis takes place basically over the ribose sugar of the PRPP, whereas in case of pyrimidine synthesis, the pyrimidine ring is synthesized first as a free pyrimidine, and then the ribose is incorporated into the nucleotide by with the help of PRPP, and UMP is generated, which is the precursor for other pyrimidine nucleotides. Is this clear to you? So in pyrimidine
Yes,
synthesis,
ma'am. I thought that the final product which is formed out of this pyramidal cycle is UMP.
so
Yeah.
I think you did not understand the question correctly,
Yes,
right?
this one.
So if it is asked like what is the precursor for
Mm.
other pyrimidine nucleotides, then UMP would be the answer.
Mm.
Okay.
Okay, ma'am.
Yeah. So moving to the next question. Which enzyme catalyzes the committed step of pyrimidine synthesis? Option A, carbamoyl phosphate synthetase 2. Option B, aspartate transcarbamylase. Option C, dihydroorotase. Option D, orotate phosphoribosyl transferase. Which enzyme catalyzes the committed step of pyrimidine synthesis? Option A, carbamoyl phosphate synthetase 2. Option B, aspartate transcarbamylase. Option C, dihydroorotase. And option D, orotate phosphoribosyl transferase. You have answered option A, carbamoyl phosphate synthetase 2. That is not the correct answer actually. The correct answer is aspartate transcarbamylase. So the committed step of pyrimidine biosynthesis is the second step. So uh, it's not necessary that the committed step is always the first step of the pathway. Okay, it's the step where all the regulation happens, where the metabolic pathway, the point where the metabolic pathway can be regulated by feedback inhibition and other mechanisms, and the point where the step where like the step of the pathway which is first of all irreversible and it is tightly regulated. So that step is the most committed step of the pathway, because if that particular enzyme is inefficient or deficient, anyway the pathway is not gonna work. Okay, so that is like the driver of that pathway. Okay, so if there's a car, the driver is the most committed point, because if there's no driver, the car is anyway not going to move, right? So this particular step would be the driver of this pathway. So in case of pyrimidine biosynthetic pathway, aspartate transcarbamylase, this enzyme, is the committed, like catalyzes the committed step of the pyrimidine biosynthesis. So the pathway starts from glutamine getting converted into carbamoyl phosphate by the enzyme carbamoyl phosphate synthetase 2, and then carbamoyl phosphate gets converted into carbamoyl aspartate by the enzyme aspartate transcarbamylase. So aspartate gets uh, attached with the carbamoyl phosphate to form carbamoyl aspartate. And this enzyme, aspartate transcarbamylase, is tightly regulated by the UTP. So UMP is the final product of this pathway, and then UMP uh, forms, and then UMP gets phosphorylated to form. Just a second. UMP gets phosphorylated to form UDP and UTP, and then UTP is converted into CTP. That is another pyrimidine. So the final product, that is UTP or CTP, all uh, show a feedback inhibition towards the enzyme aspartate transcarbamylase. So aspartate transcarbamylase is inhibited by UTP, by CTP. CTP also inhibits the enzyme that converts UTP into CTP. So these are all feedback inhibition, okay, when the product inhibits its own enzyme, synthesizing enzyme. So this is feedback inhibition loops, these are shown. And ATKs or aspartate transcarbamylase, it is tightly regulated. So when more and more product is present, the pathway signals that we don't need more UMP or UTP. So stop synthesizing more and more UTP. So what it will do, it will go and inhibit the ATKase enzyme. Once ATKase is inhibited, there won't be any formation of UMP, and then thus there won't be any formation of UTP or CTP. I hope that's clear. Let me know if you have any doubt. Huh? Okay. Moving to the next question. Defective orotate phosphoribosyltransferase transferase leads to which condition? Defective orotate phosphoribosyltransferase transferase leads to which condition? Option A, orotic aciduria. Option B, gout. Option C, Lesch-Nyhan syndrome. Option D, phenylketonuria. Defective orotate phosphoribosyltransferase transferase leads to which condition? Option A, orotic aciduria. Option B, gout. Option C, Lesch-Nyhan syndrome. Option D, phenylketonuria. Defective orotate phosphoribosyltransferase transferase leads to which condition? We are saying option A, orotic aciduria. That is the correct answer. So orotic aciduria is caused due to the deficiency or absence of orotase phosphoribosyltransferase transferase or OPRTase. Uh, that it is inherited as autosomal recessive disorder, and the clinical features are retarded growth, severe megaloblastic anemia, crystals excreted in the urine. So it is Crystalluria may cause UTI, that is urinary tract infection. Treatment is administration of UMP or uridine triacetate. So UMP is administered so that it in basically this enzyme orotate phosphoribosyl transferase you saw in this pathway, right? So this is orotate phosphoribosyl transferase converting orotate into OMP and then UMP. So if UMP is given, then it can easily be replenished. So basically, if this enzyme is deficient, there will be a deficiency of UMP. So UMP can be administered. Also, once UMP is administered, it uh, kind of regulates UMP will get converted into UTP, and UTP will regulate this pathway so that less orotate is synthesized. Okay, so that orotate accumulation will not happen, leading to orotic aciduria. Okay, so treatment with UMP is very efficient. I hope that's clear. Do you have any doubt till now? Okay, the next question is, which amino acid donates nitrogen and carbon atoms during pyrimidine biosynthesis? Which amino acid donates nitrogen and carbon atoms during pyrimidine biosynthesis? Option A, glutamine. Option B, aspartate. Option C, glycine. Option D, serine. Which amino acid donates nitrogen and carbon atoms during pyrimidine biosynthesis? Option A, glutamine. Option B, aspartate. Option C, glycine. Option D, serine. Which amino acid donates both nitrogen and carbon atoms? We are saying option A and B. So in the question, I'm asking the amino acid that is donating both nitrogen and carbon atoms. So the correct answer actually is aspartate. So if you see in the pyrimidine ring, the nitrogen, one of the nitrogen is donated by glutamine, but aspartate donates one nitrogen and the three carbon atoms. Okay, and one carbon atom is donated by the carbon dioxide. So I've told you the raw materials for pyrimidine biosynthesis is glutamine, CO2, and aspartic acid. So one carbon is coming from CO2, one nitrogen is coming from glutamine, and then rest of three carbons and one nitrogen is coming from aspartate. So in the question, if you see, I've asked which amino acid donates both nitrogen and carbon atoms during pyrimidine biosynthesis. So in that case, aspartate is the one that is donating nitrogen and carbon atoms. Glutamine is only donating the nitrogen atoms, and carbon dioxide is only donating the carbon atom. So aspartate is the one which is donating both nitrogen and carbon atom. Okay, so aspartate would be the correct answer here. Aspartate is donating the fourth, fifth, and sixth carbon atom and the first nitrogen atom. The second carbon atom in the pyrimidine ring is donated by the carbon dioxide or bicarbonate, and the third nitrogen atom is donated by the glutamine. Is this clear?
Yes, ma'am. But one assignment was given for purine synthesis. I think it is given in the bracket as multiple answers.
Okay.
In that way, we have. I mean, unless it is mentioned as multiple answers,
Yeah, and
we need
if
to choose
yeah,
only one question.
if it is a multiple answer question, it will be
Mm.
mentioned.
Yes,
Okay.
one question was there in the assignment. I think for purine ring.
Okay,
It
okay,
is the
okay.
glycine and aspartate. It was another question was asked in that assignment.
Okay, so if it is
And,
mentioned in the question that it is
the,
a multiple answer question, then you have to. But the
okay.
question asks that which
Yes.
amino acid is donating
Okay.
nitrogen
Both.
and carbon. So for that, in that case, aspartate would be
This
the
is
correct
aspartate.
answer.
Okay, ma'am.
Okay. Okay. Then moving on to the next question. Which inborn error results in the accumulation of galactose in blood? Which inborn error results in the accumulation of galactose in blood? Option A, galactosemia. Option B, phenylketonuria. Option C, alkaptonuria. Option D, maple syrup urine disease. Which inborn error results in the accumulation of galactose in blood? Galactosemia, phenylketonuria, alkaptonuria, and maple syrup urine disease. We have answered option A, galactosemia. That is the correct answer. So, by the name itself, you can see galactose in blood means galactosemia. Anything in blood, it ends with emia or semia, and if it is in urine, it ends with urea or nuria. Okay, so that's the common nomenclature. So, galactose, accumulation of galactose in blood, the condition is called galactosemia. Accumulation of glucose in blood would be glucosemia, okay, or glycemia. Okay, moving to the next question. Phenylketonuria is due to deficiency of what enzyme? Phenylketonuria is due to a deficiency of what enzyme? Option A, tyrosinase. Option B, NGPRT. Option C, glucose 6-phosphate dehydrogenase. Option D, phenylalanine hydroxylase. Phenylketonuria is due to a deficiency of what enzyme? Tyrosinase, NGPRT, G6PD, phenylalanine hydroxylase. We have answered option D, phenylalanine hydroxylase. That is the correct answer. So phenylketonuria, we have already discussed about these disorders, so I will not go into much detail. So phenylketonuria, it is caused by the deficiency of phenylalanine hydroxylase, uh, clinical manifestations, some mental retardation, hypertonia, seizures, and phenyl pyruvate is actually secreted in the urine, so it is used for the detection. So if a person is suffering from phenylketonuria, phenyl pyruvate detection in urine is a clear clinical sign. Then treatment is basically dietary restriction of phenylalanine, because phenylalanine, uh, so this enzyme phenylalanine hydroxylase, it is an enzyme in the phenylalanine metabolic pathway. So if this enzyme is deficient, phenylalanine accumulation and other byproduct formation could occur. So better to restrict phenylalanine in the diet so that the conditions or the symptoms won't develop. So phenylketonuria type 1. Moving to the next question, question 12. Maple syrup urine disease is caused by a defect in catabolism of which amino acid? Maple syrup urine disease is caused by a defect in catabolism of which amino acids? Option A, aromatic amino acid. Option B, branch chain amino acid. Option C, sulfur containing amino acid. Option D, basic amino acids. Maple syrup urine disease is caused by defect in catabolism of which amino acids? Aromatic amino acids, branch chain amino acids, sulfur containing amino acids, or basic amino acids. We have answered option B, that is branch chain amino acid. That is the correct answer. So, like phenylketonuria was a defect in the phenylalanine metabolism, maple syrup urine disease is a defect in the branch chain amino acid metabolism. So, the enzyme that is deficient in maple syrup urine disease is branch chain keto acid decarboxylase. Clinical manifestation is mental retardation is obviously there, and then along with that, maple syrup odor or a caramel-like odor is there in the urine. That's why the name. And urine secretes valine, leucine, isoleucine, and multiple keto acids. Keto acids are the one that gives the maple syrup odor. And the treatment is obviously restrict the valine, leucine, and isoleucine, all the branch chain amino acid in the diet. So, like for phenylketonuria, restriction of phenylalanine rich diet is the treatment. In case of maple syrup urine disease, valine, leucine, and isoleucine restriction is required. Okay, moving to the next question. Which enzyme deficiency leads to tyrosinemia type 1? Which enzyme deficiency leads to tyrosinemia type 1? Option A, fumarate acetoacetate hydrolase. Option B, tyrosine aminotransferase. Option C, homogentisate oxidase. Option D, phenylalanine hydroxylase. Which enzyme deficiency leads to tyrosinemia type 1? Tyrosine amino, option A, fumarate acetoacetate hydrolase. Option B, tyrosine aminotransferase. Option C, homogentisate oxidase. Option D, phenylalanine hydroxylase. You are saying option A, fumarate acetoacetate hydroxylase. Hydrolase. That is the correct answer. So tyrosinemia type 1, it is caused by the deficiency of this enzyme, fumarate acetoacetate hydrolase. Clinical manifestation again is mental retardation, hepatorenal damage. Substances in the urine found are tyrosine and PHPPA, and diet restrictions. Uh, Dietary restrictions are of phenylalanine and tyrosine. Okay. So tyrosinemia, by the name itself, tyrosine is present in the circulation, in the urine and the blood. Okay. Since we have discussed all these disorders, I'm not going into detail. So if you have any doubt, please let me know. I'll try to explain it. But otherwise, I'm not explaining it in much detail because we have already discussed it in previous sessions. So you can go through those slides if you have any doubt to clarify it. Next question is alkaptonuria is due to deficiency in which enzyme? Option A, fumarate acetoacetate hydrolase. Option B, tyrosine aminotransferase. Option C, homogentisate oxidase. Option D, phenylalanine hydroxylase. Alkaptonuria is due to a deficiency of which enzyme? Fumarate acetoacetate hydrolase, tyrosine aminotransferase, homogentisate oxidase, and phenylalanine hydroxylase. You've answered option C, that is homogentisate oxidase. That is the correct answer. So alkaptonuria, it is caused by the deficiency of homogentisate oxidase, and the clinical manifestations include arthritis and homogentisate is secreted in the urine, and there are no such treatment available for alkaptonuria. Okay. Moving to the next question now. Which condition features hyperuricemia and self-mutilation? Which condition features hyperuricemia and self-mutilation? Option A, gout. Option B, phenylketonuria. Option C, Lesch-Nyhan syndrome. Option D, galactosemia. Which condition features hyperuricemia and self-mutilation? Option A, gout. Option B, phenylketonuria. Option C, Lesch-Nyhan syndrome. Option D, galactosemia. You're saying option C, Lesch-Nyhan syndrome. That is the correct answer. So we have discussed this already. In Lesch-Nyhan syndrome, hyperuricemia occurs, and due to the uric acid accumulation in the circulation, and symptoms are, one of the major symptoms is self-mutilation, where the patient bites their own finger and scratches their own skin. Okay. Which organ is primarily responsible for the ammonia detoxification? Which organ is primarily responsible for the ammonia detoxification? Option A, liver. Option B, kidney. Option C, heart. And option D, pancreas. Which organ is primarily responsible for ammonia detoxification? Option A, liver. Option B, kidney. Option C, heart. And option D, pancreas. You're saying option A, liver. That is the correct answer. So we have already gone through the whole all the pathways, but just to summarize, so this is the metabolism of amino acids in the liver. So in the liver, the amino acids are either used up for making liver proteins, or they are used up for making nucleotides, hormones, and porphyrins, or they are transferred to the cytoplasm for making plasma proteins or tissue proteins. But along with that, they are also metabolized by deamination to form pyruvate, which can get converted into acetyl-CoA and enter the TCA cycle for ATP production, or the pyruvate can go towards glucose production by gluconeogenesis when blood glucose levels are low. And the ammonia that is released after the deamination of amino acid, the ammonia enters the urea cycle inside the liver. Other amino acids from the muscles are also transferred to the liver by the glucose alanine cycle. So if you remember the glucose alanine cycle, it is basically to transport the nitrogen or the different amino acids from the skeletal muscle to the liver, so that they can be converted into pyruvate and they can enter the gluconeogenetic pathway, or they can be used up for the ATP production. So liver basically removes most of the amino acid absorbed by the intestine. The priority uses for protein synthesis. Excess amino acids that are remaining after the protein synthesis are deaminated and converted into urea. The liver secretes about 30 gram of urea per day. Keto acids are used as fuels or for gluconeogenesis, as I've already mentioned. Okay. So we have already gone through the gluconeogenesis pathway, urea cycle, and then TCA, and how acetyl coenzyme is used for fatty acid synthesis, lipid synthesis. So this is basically a uh, integration, basically the core justification of the course name. The course name is Overview and Integration of Cellular Metabolism. So here you can see all the metabolic pathways that you have studied so far are now you can see in one single diagram, so that you can understand that everything is integrated to each other, everything is connected to each other, and everything is happening simultaneously in our body. Okay. So it's not like when gluconeogenesis is happening, fatty acid synthesis won't happen, or uh, urea cycle won't occur. So everything is happening independently, but they're happening dependent. They're also dependent on each other in a way that they are regulated by one or the other way. Okay, I hope that's clear to you. Moving to the next question. In the well fed state, the major fuel used by skeletal muscle is. In the well fed state, the major fuel used by skeletal muscle is. In the well fed state, the major fuel used by skeletal muscle is. You think option A, glucose. So in the well-fed state, the major fuel used by the skeletal muscle is glucose. That is the correct answer. So when the well-fed state means we after just after the meal, just after eating a meal. So when we have taken a meal, immediately after that, the blood glucose level spikes because all the carbohydrates that we have eaten, all the other metabolites that we have eaten, everything is getting converted into glucose, and glucose levels are high. And since the glucose levels are high, all the organs start using the glucose for energy production. So as soon as we eat food, the glucose level goes high, and the glucose a high glucose level immediately triggers the insulin secretion, and the insulin secretion basically signals uh, the liver to send the glucose sorry, insulin secretion basically signals all the organs to take up the glucose, where basically promotes the glycolytic pathways. So glucose is converted into ATP. So muscle also takes up the glucose. So in usual cases, muscles generally don't use the blood glucose. Blood glucose is mostly spared for the organs like brain and RBCs, which require more energy and they require constant energy. So they mostly use the glucose directly, and the skeletal muscle and all they use other sources, which are other sources of ATP production like fatty acids and ketone bodies and etc. So blood glucose is generally used by organs like brain and RBCs. So, but just after eating a meal, which is the well-fed state, the absorption of glucose from gut occurs and blood glucose level spikes. So that uh, triggers the use of glucose for triacylglycerol production, for ATP production in the skeletal muscles, and all the other pathways. So insulin secretion basically suppresses all the pathways that are going towards the glucose formation, like gluconeogenesis and catabolism of fatty acids and such, etc. So, and it triggers the pathways that are promoting the glucose breakdown into energy. Okay. Is that clear?
Yes, ma'am.
Moving to the next question. During prolonged starvation, which organ still requires glucose? During prolonged starvation, which organ still requires glucose? Option A, liver. Option B, brain. Option C, heart. Option D, kidney. During prolonged starvation, which organ still requires glucose? Option A, liver. Option B, brain. Option C, heart. Option D, kidney. You're saying option B, brain. That is the correct answer. So, as I've mentioned in the previous slide, also, brain uses the glucose as the main ATP source. Since fatty acids cannot cross the blood brain barrier, so glucose can easily enter the brain and it can be converted into ATP. Uh, Brain constantly requires energy. Brain is the major control unit of your body. So, for constant energy production, brain uses the glucose even when prolonged starvation cases are there. So, when prolonged starvation occurs, all the glucose that is present, that is coming from gluconeogenesis, and whatever glucose is spared by the skeletal. So none of the other organs uses glucose as the fuel source. They use other fuel source like fatty acids and ketone bodies, but brain uses the all the glucose that is present in the blood because fatty acids cannot cross the blood brain barrier. Okay. Next question is, what is the main energy source for muscle during overnight fasting? What is the main energy source for muscle during overnight fasting? Option A, glucose. Option B, fatty acid. Option C, ketone bodies. Option D, glycogen. What is the main energy source for muscle during overnight fasting? Option A, glucose. Option B, fatty acid. Option C, ketone bodies. Option D, glycogen. What is the main energy source for muscles during overnight fasting? Glucose, fatty acid, ketone body, glycogen. You have answered option D, glycogen. After overnight fasting, I've told you the muscles actually do not depend. So, uh, according to your answer, glycogen uh, as an energy source means that glycogen will be broken down into glucose and then glucose will be used as energy source, right? But I've mentioned that all the glucose that is uh, formed by glycogen breakdown or by gluconeogenesis, all the glucose is actually spared in case of starvation. It is spared for organs like brain. So in such cases, in cases of starvation, in cases of fasting, muscles and other organs which can use other energy sources, they generally rely on that. So in this case, muscles uses fatty acids as the energy source. So this is a like different fuel sources are used by different organs. So you can see after a meal, glucose is used up by almost all the organs, okay? Except adipose tissues. Adipose tissues also use glucose, but mostly since they have a lot of fatty acids, they use the fatty acid as the major source. But after a meal, since glucose is in excess, all the organs use glucose as the major fuel source. Okay. But after short-term fasting, like overnight fasting, brain still uses glucose as the fuel source because it has no other options. The other option is ketone body, which it uses after long-term fasting because ketone bodies can cross the blood-brain barrier. Fatty acids cannot. Okay. So after short-term fasting, all the glucose that is there is spared for the brain. Skeletal muscles, cardiac muscles, adipose tissue, they all rely on fatty acids. After long-term fasting, brain relies on glucose and ketone bodies. Skeletal muscles also rely on ketone bodies and sometimes branched chain amino acids. Cardiac muscles rely on ketone bodies. Also, the adipose tissue, they also rely on ketone bodies and fatty acids. So, major fuel sources for organs are glucose, fatty acids, and ketone bodies. Okay. And from that, glucose mostly is used by brain. Okay. Other muscles can rely on fatty acids and ketone bodies, but brain has to rely on glucose. So after a short term fasting, skeletal muscle uses the fatty acids, and whatever glucose is present is always spared for the brain to use. Okay, because brain cannot use fatty acid. Is that clear?
Yes, ma'am. But glycogen stores will be it's given ma'am,
So
so it's depleted.
glycogen stores would be used up. It will be used to convert glycogen into glucose, and then glucose will be transferred to the brain. Muscles won't use the glucose for ATP production. Muscles will use the fatty acid for ATP production. The glucose that will be formed after glycogen breakdown in the muscle will be transferred to the brain because brain needs the glucose first. Okay.
Okay, ma'am.
Mhm. Okay, moving to the next question. During starvation, the which pathway is upregulated to provide glucose? During starvation, which pathway is upregulated to provide glucose? Glycolysis, option B, gluconeogenesis, option C, glycogenesis, option D, pentose phosphate pathway. During starvation, which pathway is upregulated to provide glucose? Option A, glycolysis. Option B, gluconeogenesis. Option C, glycogenesis. Option D, pentose phosphate pathway. We are saying option B, gluconeogenesis. That is the correct answer. So all the other pathways, glycolysis is to utilize glucose and convert it into pyruvate. So it's a glucose breakdown pathway. Glycogenesis is again use of glucose to make glycogen. Pentose phosphate pathway is again it uh, glucose is converted to glucose 6 phosphate, which enters the pentose phosphate pathway. So all the other pathways are basically the pathways where glucose is used up, whereas gluconeogenesis is the pathway where glucose is formed. So during starvation, uh, gluconeogenesis is upregulated because since during starvation, blood glucose level will go down. to maintain the blood glucose level, gluconeogenesis will help convert all the pyruvate to get converted into glucose so that the glucose can be transferred to the blood from the liver. Okay, so this is the early fasting stage where the gluconeogenesis is upregulated. So pyruvate is converted into glucose 6 phosphate and then glucose, which is transported to the brain and the circulation. And other than that, so I told you during well-fed state, the hormone that uh, is stimulated is insulin. During the fasting state, uh, glucagon is stimulated, and glucagon triggers basically the upregulation of gluconeogenesis. So this is called the post-absorptive state. That is within about 1 hour of meal, blood glucose level begins to fall, and by 2 hours, it reaches the normal level. Blood sugar level maintained by hepatic glycogenolysis. So glycogenolysis is basically breakdown of glycogens into glucose. Neoglucogenesis or gluconeogenesis, which is the conversion of pyruvate into glucose. Lipolysis, that is the breakdown of lipids into glucose, fatty acids or lipids into glucose. So this is the lipolysis part. Then glucose sparing action of skeletal muscle and adipose tissue, which I told you that in case of starving, skeletal muscles and adipose tissue, instead of using the glucose, they use the other sources like fatty acids and ketone bodies for energy sources, and they spare the glucose, whatever glucose is remaining, only for the brain. Okay, like in the previous question, I have told you glucose sparing action of skeletal muscle and adipose tissue so that brain can use all the glucose that is available, because that is in that case brain requires glucose the most. Okay, other tissues have other options as well, but brain does not have many other options. Brain can either use glucose or in extreme cases it can use ketone bodies. Okay, so these are the effects of glucagon on the blood glucose. So glycogen breakdown is upregulated, that is glycogenolysis. Glycogen synthesis is downregulated so that less glucose is stored as glycogen. More glycogen is broken down into glucose because at that time we don't have enough glucose. So glycogenesis is downregulated. Glycolysis is downregulated in the liver so that less glucose is used as fuel. Gluconeogenesis is upregulated so that more and more uh, glucose can be formed. Fatty acid mobilization is upregulated, less glucose is used as fuel, and ketogenesis is upregulated so that it provides an alternative to glucose as energy source, so that the organs which are not getting glucose as the fuel can get ketone bodies as the fuel. And the target enzymes are mentioned here. So for glycogen breakdown, glycogen phosphorylase is upregulated, glycogen synthase is downregulated. Uh, in case of glycolysis, PFK1 is downregulated, FBPase2 is upregulated, pyruvate kinase is downregulated, DP carboxykinase is upregulated, then triacylglycerol lipase is upregulated, acetyl coenzyme carboxylase is also upregulated. Okay, so these are all the metabolic effects and the enzymes that are affected by the glucagon. So glucagon triggers all these pathway, the upregulation and downregulation of all these pathways, depending on the blood glucose level. When blood glucose level is high, insulin is secreted, and the opposite metabolic effect is seen. When blood glucose level is low, glucagon is secreted, and glucagon triggers all these metabolic effects. Okay, I hope that's clear. Moving to the next question. Which hormone is increased during fasting? Option A, insulin. Option B, growth hormone. Option C, thyroxine. Option D, glucagon. During fasting, which hormone is increased? Option A, insulin. Option B, growth hormone. Option C, thyroxine. And option D, glucagon. We have already discussed this, so you can answer. Which hormone is increased during fasting? Yes, option D, glucagon. So I've already told you glucagon is increased during fasting. Next question, which tissue is the main site of ketone body synthesis? Which tissue is the main site of ketone body synthesis? Option A, liver. Option B, kidney. Option C, muscle. Option D, adipose tissue. Which tissue is the main site of ketone body synthesis? Option A, liver. Option B, kidney. Option C, muscle. And option D, adipose tissue. You are saying option A, that is liver, that is the correct answer. So we have already discussed ketogenesis, how ketone bodies are synthesized, what are ketone bodies. So no need to discuss it again. I've just added the questions so that you will have an idea what kind of questions can come in your exam or can be there in your assignments. The next question is, what is the principal fuel for cardiac muscle during starved state? What is the principal fuel for cardiac muscle during the starved state? Option A, glucose. Option B, fatty acid. Option C, glycogen. Option D, amino acid. What is the principal fuel for cardiac muscle during starved state? Glucose, fatty acid, glycogen, or amino acid? We are saying option B, fatty acid. That is the correct answer. So I've told you previously also, skeletal muscle and cardiac muscle, they use fatty acid as a fuel so that glucose can be spared for the brain. So metabolism in the cardiac muscle or in the heart, it is continuously active in regular rhythmic contraction and relaxation. You know that heart is continuously working, right? Cardiac muscles are continuously working, unlike your other skeletal muscle, which are uh, voluntary muscle. This is an involuntary muscle, right? So it is working irrespective of you want or not, don't want it to work, right? Like you have a control on your hand. If you don't want to move your hand, you can voluntarily stop moving your hand. But then cardiac muscle is an involuntary muscle. It will move on its own and it will have a continuous rhythmic motion. So it obviously requires fuel, and in that case, it uses a variety of fuel, but mainly it uses fatty acid. It uses glucose, lactate, ketone bodies also. It uses glucose uh, when we are at the well-fed state, but mainly it uses the fatty acids so that the fatty acids can be converted into ATP, and that ATP can be used by the cardiac muscles for the motion. Okay. Metabolism of heart muscle differs from that of the skeletal muscle in three important respects. First is variation in the work output is far less than that seen in the skeletal muscle. So, skeletal muscles, since although they are voluntary muscles, they have a very varied output, right? Like you can run with your legs, you can walk with your legs, you can choose not to do anything, sit relaxed. So your skeletal muscles have different variations in its function, right? But heart muscles function is very rhythmic, very regular. It's not It's not like your heart muscles are one day it is contracting and relaxing very fast, and second day it is doing it very slow. It's not like that. It's a rhythmic motion. It's going on continuously ever since you are born. It's the same. Heart is completely aerobic tissue, whereas skeletal muscles can function anaerobically for limited periods. So, if you remember, we have studied the lactate formation from the pyruvate, which was the anaerobic respiration by the muscles. So, when you do very vigorous exercise and the oxygen limits, the oxygen is not present, like it doesn't reach the muscles for the electron transport chain to occur. In such cases, the pyruvate gets converted into lactate, which is the anaerobic respiration pathway. Uh, so, mitochondria are much more densely packed in heart than in other cells. So, in case of heart, no such anaerobic respiration occurs. There are a lot of mitochondria present in the heart, and they basically are always present, and oxygen is always available to the heart. Once oxygen cutout occurs in the heart will immediately stop working. Okay. And the third point is heart contains negligible energy reserves as glycogen or lipids, although there is small amount of creatine phosphate. So like muscles have the option of keeping glycogen, liver has the option of keeping glycogen, other tissues have the option of keeping fatty acids. Heart does not have any storage units. So heart continuously uses the fatty acids to make ATP, and the fatty acids are in the circulation. So it takes the fatty acids from the circulation or glucose from the circulation and uses it immediately for making ATP so that it can contract and relax. Okay. And supply of both oxygen and fuels from the blood to the heart muscle must be continuous to meet its energy demand, which I have already explained. Okay. So, and this also I have already shown that cardiac muscles, just after a meal, it can use glucose or pyruvate from the circulation for energy sources as a fuel source. But short after a short term fasting, it uses fatty acids and spares the glucose for the brain. Okay. Is this clear?
Yes, ma'am.
Next question says, in the starve feed cycle, insulin level rise after eating and lead to which process? In the starve feed cycle, insulin level rise after eating and lead to which process? Option A, glycogenolysis. Option B, ketogenesis. Option C, lipolysis. Option D, glycogenesis. In the starve feed cycle, insulin level rise after eating and lead to which process? Option A, glycogenolysis. Option B, ketogenesis. Option C, lipolysis. And option D, glycogenesis. In the starve feed cycle, insulin level rise after eating and lead to which process? Option A, glycogenolysis. Option B, ketogenesis. Option C, lipolysis. Option D, glycogenesis. You are saying option C, lipolysis. What do you think? After eating, insulin level rises. Okay. Why is the insulin level rising? Rise of insulin level triggers towards what? Increase in glucose synthesis or decrease in glucose synthesis? Think of it in that way first. So we have just eaten the food. So now, what do you think will happen? Body will trigger for making more glucose, or body will trigger for utilizing the glucose? First answer that. Just after eating food, what do you think will happen? Body will try to utilize the glucose, or body will try to make more glucose?
will try to utilize this.
Utilize the glucose, right? Because now
Yes,
we have
ma'am.
eaten, so we have already eaten a lot of carbs and there is a lot of glucose. So if body starts making more glucose, there will be high blood sugar, right?
Oh,
So it will try to utilize the glucose, and that's why insulin level rises so that it can trigger all the pathways that will utilize the glucose, right? Now think of, now look at all the options: glycogenolysis, ketogenesis, lipolysis, and glycogenesis. Which option do you think utilizes the glucose? What happens in,
glycogenesis,
yes, glycogenesis.
glycogenesis.
Yes, glycogenesis is the pathway where what happens?
Glucose will be utilized for storage purpose. It will be
Yes,
converted to
yes,
glycogen.
yes. So glucose will be utilized. In all the other cases, like glycogenolysis, glycogen is broken down into glucose. Ketogenesis, ketone bodies are used up for fuel. Lipolysis, lipids are broken down for fuel. Only in case of glycogenesis, glucose will be used up. So insulin
Utilized.
will trigger all the pathways where the glucose can be used up. Okay. So the correct answer will be option D.
Okay, ma'am.
Okay. So these are all the metabolic effects of insulin, like you saw in case of glucagon. So all these things will be opposite of glucagon influence. So in case of insulin, obviously, as I've told you, glucose uptake will be upregulated in the muscles and adipose tissue and in the liver. Uh, Glycogen synthesis will be upregulated in the liver and muscle. Glycogen breakdown will be downregulated. Glycolysis, acetyl CoA production will be upregulated in the liver muscle. Fatty acid synthesis will be upregulated. So fatty acid synthesis will be upregulated in the liver. Fatty acid breakdown will be downregulated. Okay.
Okay, ma'am.
So similarly, all the synthetic pathways will be upregulated with insulin secretion or just after eating. Like think logically, like when you are eating, that means you already have enough glucose. So at that time, your body will try to use all the glucose for other metabolic synthesis. Okay, it will use the glucose for glycogen synthesis. It will use the glucose for fatty acid synthesis. Okay, and then when after that, when you have stopped eating, when you are not eating for a long time, when there's a period of starvation, in that part, the body will now use all the synthesized product to again either make glucose from it or use it as a fuel source or use it as a energy source. Okay, so all this is very logical. You don't have to mug it up like, okay, insulin is upregulating glucose uptake. You can logically think like, what happens? Why do you think insulin is given to a diabetic patient? Insulin is given to a diabetic patient because insulin triggers all the glucose uptake pathways. Okay, so in a diabetic patient, you know that the blood sugar level is high, the blood glucose level is high. So if we inject insulin from outside, the glucose will be utilized and the blood glucose level will go down. Okay, so think logically. All these things are something that you know. You just have to read the question properly and answer. Okay. Moving to the next question now. Deficiency of glucose 6 phosphatase causes which inborn error? Deficiency of glucose 6 phosphatase causes which inborn error? Option A, Von Jerky's disease. Option B, McArdle's disease. Option C, Pompe disease. Option D, Cori disease. You're saying option A, Von Jerky disease. That is the correct answer. So, von Wiener's disease is caused by the deficiency of glucose 6 phosphatase. So, glucose 6 phosphate cannot be converted to glucose, and this glucose 6 phosphate will enter the HMP shunt pathway. And as a result, ribose 5 phosphate, that is the pentose, the HMP shunt pathway is basically the pentose phosphate pathway. So the pentose sugar will be high in circulation, and you know that the new in the nucleotide synthesis, in the purine synthesis pathway, we saw ribose 5 phosphate is used for the PRPP synthesis. So if ribose 5 phosphate is high, then amount of PRPP will go up, and amount of PRPP going up means amount of purines going up. Amount of purines going up, that means the catabolic pathway of purine. If you remember the catabolic pathway of purine, where purine was converted into xanthine, hypoxanthine, and finally to uric acid, that will also increase. So ultimately, in case of von Jurke's disease, due to the deficiency of glucose 6 phosphatase, one enzyme which is converting glucose 6 which is basically removing the phosphate group from glucose 6 phosphate to make it glucose, converting glucose 6 phosphate into glucose, that step is hampered, and due to that step, all the other pathways that are connected to glucose 6 phosphate, that is the HMP shunt pathway, the purine biosynthesis pathway, the uric acid synthesis uh, from the degradation of purines, all those will be affected, and then ultimately uric acid amount will increase in the circulation. It will be excreted, resulting in uh, uric acid written sorry uric acid will be increased in the circulation, resulting in hyperuricemia. Uh, Glucose 6 phosphate gets converted into lactic acid, which will compete with the uric acid for excretion, resulting in uric acid retention. Okay. So now you see how all the pathways are connected here, right? So HMP shunt pathway, we read about HMP shunt pathway when we were talking about carbohydrate metabolism. But now you see it is also connected to the nucleotide metabolism. Okay. So everything is interconnected to each other. Okay, so you have to understand all the metabolic pathways that we have been studying since 10 sessions, and then the next two sessions, the 11th and the 12th session, is all about integrating all the pathways that you have studied into one big picture. I hope this is clear to you.
Yes, ma'am.
Moving to the next question now. A defect in carbamoyl phosphate synthetase 1 causes option A hyperammonemia, option B hypoglycemia, option C metabolic acidosis, option D gout. A defect in carbamoyl phosphate synthetase 1 causes option A hyperammonemia, option B hypoglycemia, option C metabolic acidosis, option D gout. A defect in carbamoyl phosphate synthetase 1 causes option A hyperammonemia, option B hypoglycemia, option C metabolic acidosis, option D gout. You're saying option A, hyperammonemia. That is the correct answer. So there are two carbamoyl phosphatase and phosphate synthetase in the body. One is CPS1 and one is CPS2. CPS1 uses NH3, that is the ammonia, as the nitrogen source, and which is a part of urea cycle. And CPS2, as you have seen, it is involved in the pyrimidine biosynthesis pathway, where it uses glutamate as the nitrogen source. Okay. So CPS1 uses ammonia as the nitrogen source, and if CPS1 is defective, ammonia will be accumulated in the circulation, leading to hyperammonemia. I hope this is clear. We have already discussed urea cycle, so in that part we had talked about hyperammonemia. So I hope all this is clear. I'll move on to the next question. Which enzyme is allosterically activated by ATP during pyrimidine synthesis? Which enzyme is allosterically activated by ATP during pyrimidine synthesis? Option A, CPS2. Option B, aspartate transcarbamylase. Option C, dihydroorotase. Option D, thymidylate synthase. Which enzyme is allosterically activated by ATP during pyrimidine synthesis? Option A, CPS2. Option B, aspartate transcarbamylase. Option C, dihydroorotase. And option D, thymidylate synthase. Which enzyme is allosterically activated by ATP during pyrimidine synthesis? Option A, CPS2. Option B, aspartate transcarbamylase. Option C, dihydroorotase. And option D, thymidylate synthase. You're saying option A, that is CPS2. Think again. I have already told you which. Acha, tell me which enzyme was the with which enzyme was catalyzing the committed step of pyrimidine synthesis. I've told you which enzyme is tightly regulated in the pyrimidine
This
synthetic pathway.
aspartate transcarbamylase.
Yes. So that is the enzyme that is activated and inhibited allosterically so that the whole pathway can be regulated. So the correct answer is aspartate transcarbamylase. Okay. So don't just guess the answer. You have to think. I've already discussed this whole regulation, right? So ATKs, it is along with the feedback inhibition. So uh,
It's activated by AMP and GMP, no, ma'am.
it
But
is huh, it is inhibited by UTP, CTP, and it is activated by ATP. Okay.
EDP and GT.
And
Okay.
in this case, also CTP synthetase is also activated by GTP. Okay.
GTP.
So you have to think the steps where the regulations are happening. Okay. In the pyrimidine biosynthesis pathway, the major regulation site is ATKs. Okay. So with this, we come to the end of this session. If you have any doubt in any of the questions discussed or any of the topic that is there in this week's session, let me know. We can have a discussion. We have enough time. Do you have any doubt?
We will have one more session, ma'am. Live session next week, or this is the last session, ma'am.
We have one more session. So this was the 11th
Next.
session, right? Yeah. For the 12th
Okay.
week, next Tuesday,
Okay.
we'll have one more session. That would be the last session. Okay. So
Okay,
if you
ma'am.
have any doubt in any of the topics from the previous sessions also, you can ask in that session. So
Yes,
I'll
ma'am.
keep all the slides in my laptop ready so that whichever where if you have any doubt in any of the previous sessions question also let me know. I'll open it and I'll try to solve it. Okay.
Okay, ma'am. Thank you, ma'am.
Yeah. Thank you. Thank
Thank
you
you,
so much
ma'am.
for joining. I'll then end this session if you don't have any doubt. Thank you. Bye bye.